Good morning, everyone. We're live. So, everybody say hi.
Oh, good morning.
All right. So, exam-wise, I had at one point a vague notion, what was it saying, that we would have the exam real-time in person. I think what we're going to do is on a day, probably next Friday, we won't meet. What we'll do instead is at 8 a.m., I will drop the exam on Moodle, and then at 10 a.m. it'll be due. And so that means it's completely open note, open book. You should gather with your smart friends and ask them everything. No, you need to work independently. But it'll be 2 hours, and the exam is designed to be taken in class in an hour. And we'll see how that goes. It's not like my favorite thing. I worry a little bit about, you got to have some internet connection. Certainly feel free to just sit right here and do it or be on campus. Anyway, we'll work out the specific details in terms of exactly the date, but I'm thinking that it's going to be next Friday. And my plan at this point is, at least for this first exam, we'll do a 2-hour window. And partly, Moodle sucks. And the kinds of tests format that Moodle does is just not what I do. And so I'm actually going to give you a Word file that you can either fill out online, or you can print it out and scan it in, or heck, you can print it out and I'll be standing around at 10, and you can turn it in. So a lot of ways we can do this. Think about it. If you see problems that I don't see yet, holler.
Will there be things that we need to draw where we should print it out?
Yeah, there might be. I actually probably I may have to rethink things a little bit because of that. Because I do love saying draw two amino acids linked through a peptide bond, but I may change that question up. So yeah, this is an experiment. If it sucks, well then we'll probably do it again, just because Anyway, let's see how it goes, and we're also going to have a transcription translation homework assignment that I'm going to put out that'll be due after the exam, so I don't want anybody to be pressured or anything. And that's going to be counting towards sort of this extra points that are essentially another exam worth of credit, and it's mostly completion stuff. You do it, you get the points. All right, so that's what I'm thinking about, and we'll see where we go. All right, so it's warming up. It's amazing. Enzymes are able to actually carry out their biochemistry. We have little microbial friends outside that are singing their little happy dance, or doing their happy dance, whatever bacteria do when it's warm. They're dividing. All right, so if you watched the video, and I hope you did, we've been getting into the mechanisms of gene expression. And we established initially what we refer to as the molecular dogma. That is the flow of information from DNA. DNA is transcribed to RNA. RNA is translated into protein. And we're going to get into protein translation in a bit. Right now what we're talking about is the production of RNA. And so we can define a gene, I think, fairly well as being a piece of DNA that is able to be transcribed, or that is transcribed. I guess you could force pretty much any piece of DNA to be transcribed by manipulating it, but regardless. So we have the structural gene. That is the portion that is transcribed. That's what we designate as +1, is the first nucleotide of transcription. And then there is going to be a termination site, which would be the last nucleotide of transcription. The critical part, though, is the control elements. And this is the realm of the terms that we have talked about, specifically the promoter. And can you see that? Yeah, reasonable enough contrast. The promoter. The promoter is a DNA sequence that is adjacent to and upstream of +1. So the promoter, we're going to just drag our little box here. And then here is +1, This is the direction of transcription. This is the piece of DNA that's going to get transcribed and turned into an RNA. 5' to 3'. And as we were talking about with the topology of these double strands, this RNA sequence is equivalent to the top strand. Just that T is now U, but that's really irrelevant. So that's the orientation we're talking about. Now the promoter, what makes the promoters do its thing? This is, as all these things are, a DNA sequence. And enzymes, RNA polymerases, have a binding affinity to a promoter. So promoters end up being a fairly constrained set of sequences, what we refer to as the consensus sequence. And so we can identify by looking at a bunch of promoters what is sort of the standard or canonical best fits promoter sequence. And RNA polymerase interacts with this. So we'll talk a little bit more later about DNA binding proteins, and probably after the exam. Regardless, DNA binding proteins typically have residues, amino acid residues, that are carrying a positive charge, positive R groups. They interact with a negative charge. And they really do have sort of extensions that are able to sort of feel along the DNA and actually identify base pairs. And there is a base pair sequence that the RNA polymerase is going to interact with. I mean, it is going to be a nucleotide sequence, like 20 or so nucleotides. That's what this protein binds to. And so it has a very high affinity. It's got a very high specificity. Now, we start talking about control of gene expression, how we turn genes off and on. And this provides us actually with some really interesting comparisons between the prokaryotic bacterial model, which is pretty simple, and the eukaryotic, remember you are a eukaryote, the more complex model. And one of the key points and key differences is the bacterial RNA polymerase is made up of the alpha 2 beta prime, 4 proteins, 2 identical alphas, 1 beta, and 1 very similar to beta, but it's actually a different protein. And this is able to carry out the polymerization of an RNA molecule. It's able to read the sequence. It's able to carry out by itself what is known as the core enzyme. It can carry out the transcript elongation. What it can't do is bind to promoters and carry out transcript initiation. So we have a lot of alliterative terms of initiation, elongation, termination, all across the nation. And I was going to say something that would rhyme or bring in the idea of urination, but I decided not to do that. Transcript elongation is what the core enzyme can do. In order to have transcript initiation, we have to have what is now called the holoenzyme. And that has the alpha 2 beta prime sigma factor. So sigma factor is what controls specificity, as it turns out. So in the prokaryotic world, RNA polymerase has an intrinsic sort of just set up binding affinity to DNA. If you wanted to purify RNA polymerase from bacteria, you'd build yourself a little affinity column. You take little beads of sand, you attach just random chunks of DNA, and you pass extracts of bacteria over this. RNA polymerase would bind. You can knock it off with a little bit of salt, and you can purify RNA polymerase. Now, in order to bind specifically to the promoter, we have to have sigma factor. And so the general story is the holoenzyme binds to the promoter. Core enzyme without sigma factor is either in the process of elongation or is just randomly hanging out at different spots, not doing anything. But when we have sigma factor sitting in there, when we have sigma factor sitting in there, it now has binding affinity specifically to promoters. And we'll see, this is a little point to keep in mind. You can have promoters that are not consensus sequences. They have actually a few nucleotides off from the best fit. And this is a control system in which you may need another protein to help it bind. That's like one of the control things that bacteria can do. But the main point is that sigma factor is critical for promoter recognition. The core enzyme is exhibiting what's known as loose binding or non-specific DNA binding. Holoenzyme exhibits very specific binding to promoters. The holoenzyme binds, and specifically we were looking at some of the sequences. You don't need to really memorize them, but there is a -35 sequence, the cat box. There is the -10 sequence, that is the Tata box, or sometimes it's TA. It is an AT-rich region that you can open up, you can melt in, and you can begin reading the bottom strand and generating a new top strand. That is polymerization of RNA. Now, the RNA polymerase proceeds, and then there are a variety of termination systems. And again, we get more into some of the termination mechanisms in molecular genetics. From the perspective of protein translation, termination is kind of like, yeah, whatever, stop whenever you feel like it, because the protein coding region is going to be embedded in the middle of the transcript. It's not going to be at the very end, or it's not going to be at the very beginning. And so you got slack. That is, you have nucleotides that are actually not part of the coding region. And so if you put them in, fine. If you decide to drop a few out, that's fine too. It is the middle part that you got to make sure you get every single nucleotide right, because that's what we really translate. So termination, it happens, but I don't get overly concerned about it. When we're doing molecular genetics projects and building new genes, sometimes we will use a terminator that happens to be embedded in sequence. Other times you just figure there's a termination site somewhere out there, it'll stop, that's fine. All right, so termination, we don't really worry about. Initiation is the really cool part. And because sigma factor provides specificity, one of the interesting things that we see in terms of bacterial gene expression, and I'm just looking to make sure I do call outs to any sort of relevant figures and things. Blah, 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 blah. Yeah. All right, so looking at figure 7-9 is a nice one. That's showing us where signals in the nucleotide sequence of a gene tell the bacteria RNA polymerase where to start and to stop. You got the promoter, terminator. And we'll get into this a bit more when we start talking about gene regulation. But one of the things that bacteria can do is something called sigma factor swapping or sigma factor switching. In that when bacteria are under sort of standard conditions, they've got a specific sigma factor called sigma factor 70, not anybody cares, that is driving and pointing the RNA polymerases to standard promoters to produce all the genes you need for just standard operation. Under times of stress, like dehydration or potentially high temperature, you actually have a sigma factor swap out. A new sigma factor will come in, and it'll direct the RNA polymerase to bind to a set of different promoters and actually begin putting on genes that are used for stress. So sigma factor switching is a pretty cool thing. Our gene regulation is a good bit more complicated, but for bacteria it works fairly well. All right, so we'll be talking about some regulatory stories in the next chapter. Now, once the RNA polymerase has started up, that is, initiation is complete. And once it kind of gets down the road, about 50 nucleotides or so, the sigma factor diffuses away and goes and finds another core enzyme to interact with, and the core enzyme is able to continue on. Now, one of the cool things about this is that once the RNA polymerase has sort of cleared the promoter, and you get about 50 nucleotides in, another RNA polymerase can jump right on in and start its own transcription. And so we end up at any one point with RNA polymerases that are down here having produced almost the entire RNA. Some RNA polymerases are here, having produced half of it. Some RNA polymerases are just starting out. And so you have a whole range of transcript sizes coming off, and this thing is being filled up. It looks almost like a Christmas tree. So, and there's various diagrams and things that are shown, electron micrographs, that we interpret like this, where you have the promoter sequence here, and then here's the structural gene, and then using some fairly sophisticated electron microscopy techniques, you can see something that looks like this. It looks a little bit like feathers coming out. And that this is the RNA transcript, and the further down it gets, the longer the transcript is, and that's because we have initiation and the guy just starting out, and as soon as he clears, the next guy can jump in. So that is the process of transcription in the prokaryotic world. And yeah, that's really about all I have to say about that. Okay, so now, when we start thinking about the eukaryotic world, oh honey, things are a lot more complicated. And let's face it, not surprisingly, a bacteria, if you look at them under a microscope, A, they teensy little fellas. B, they're a dot. They divide, they're another dot. They don't differentiate particularly. I mean, there's some that will sporulate and stuff, but they're just dots. And that's how they live their lives, is just dots. They just divide. And so single-celled organisms, particularly like bacteria, they live in a really simple kind of world. He's a dot, he divides, this one dies, that one's the same thing, it's all clonal, nobody cares. They don't do a whole lot of fancy gene regulation. You know, like there may be some conditions that start to change. There may be, they run out of their favorite food, glucose, and they have to shift to something they don't like as much. But, really they don't do that much. With eukaryotes, and particularly with multicellular eukaryotes, we are doing a whole bunch of things. And one of the key points here is multicellular eukaryotes. Like you and I, we are multicellular, but we ain't all the same cell type. I got some liver cells, I got some nose cells, I got toe cells, I got hair cells. I got all kinds of different cells. And different cells are expressing different genes. So that means that the control and regulation of genes in multicellular eukaryotes is a lot more complicated, and B, pretty darn critical, because you don't want to have a liver gene being expressed in your neurons. That could be a bad deal. You need to have very tight control over genes that are triggering cell division, because rampant cell division, that's cancer, tumor city. You don't want that. So the control of gene expression in eukaryotes is a gigantic book. The control of gene expression in prokaryotes is a very small book. All right, so we have covered our prokaryotic friends, and it's not too complicated. The main point is that proteins like RNA polymerase have an intrinsic binding affinity, and when the sigma factor is present, they now have a specific binding affinity to sequences that we call promoters. Okay, so now we start talking about the eukaryotic world, and you can spell it with a C or a K, whatever. The eukaryotic world is a little bit more complicated in that, first off, we've got multiple RNA types that are going to be regulated and operating in a different way. So the multiple RNA types are pretty easy to list. We have ribosomal RNA, or RNAs. These are the RNAs that make up the ribosome. The ribosome is a ribonuclear protein. It's got a great big RNA, and then there's a bunch of proteins kind of stuck onto it. So ribosomal RNAs are produced at a really high level. Just as a little point, if you recall, this spot in the nucleus called the nucleolus. The nucleolus is a little, it's like a little bubble in the nucleus. This is a site of ribosomal RNA production. I mean, it's not really an organelle. It is just a point, a spot where the chromosomes are, or the chromosome is, that's got the ribosomal RNAs. They're producing so much RNA that they actually make a little discontinuity in the nuclear cytoplasmic goo. And you have this little bubble that we call the nucleolus. It is simply the site of ribosomal RNA production. If you were to extract RNA from yourself, your roommate, your plant, eukaryotes typically have 90% of the RNA is ribosomal RNA. I mean, that's really what you're made up of. Now, the other RNA class is the class of RNAs that can be translated, what we call the messenger RNAs, or mRNA. As a developmental geneticist, really this is the only RNA that I'm interested in. Ribosomal RNAs are fascinating, I'm sure. They generate the structure for the ribosome, very important, but every cell produces the same type. Messenger RNAs, now this is where all the fun stuff is. Messenger RNAs, we can lump them into this notion of the housekeeping genes. Messenger RNAs that are present in every cell, and then we have the really cool RNAs. These are cell-specific RNAs. It'd be interesting to know what is the messenger RNA population in a neuron that is different than the messenger RNA population in the liver. You can begin to get at what makes a neuron a neuron. And so that actually is a whole area of study of looking at differential RNA expression between different cells and learning how that works. So messenger RNA. The third type of RNA and this list is both for prokaryotes and eukaryotes, but prokaryotes are generating all these RNAs with the same RNA polymerase. Eukaryotes have different ones. The third type is transfer RNA, or tRNAs. Actually, I think we threw this chart up on Monday. So tRNAs are small, 200 or so nucleotide RNAs that fold up into this very specific structure, and they allow for the translation of an RNA into a protein. They're the actual carriers and holders of amino acids. We'll talk about that later. All right. So now, in the prokaryotic world, all these RNAs are produced by the same RNA polymerase. Just RNA polymerase, alpha 2, beta, beta prime. In the eukaryotes, we got a little bit of subdivision. We got some different guys in that we have 3 different RNA polymerases. Given the very creative name of Paul 1, Paul 2, and Fred. No, Paul 3. So these are the 3 different RNA polymerases, and each one is made up of a bunch of individual proteins. It's a great big protein complex. Just from a molecular genetics perspective, people that work on Paul 1 genes and Paul 3 genes, I don't want to be mean, but they're pretty seriously nerdy. I mean, it's like, really? That's what you work on? This is where the interesting stuff is. Paul 2 genes. These are the genes that are going to get translated. I mean, so that's what, interesting people work on. Not that there's anything wrong. If you decide to go and get a PhD and you're working on Paul 1, go for it, man. Yeah, whatever. So Paul 2 has got some really cool regulation because this is where we're going to be turning on specific genes for different cell types. All right. So the key difference then between eukaryotic RNA polymerases and prokaryotic RNA polymerases is actually an interesting and fundamental point in that these have no DNA binding affinity. Which, if you think about this for a moment, it's like, what the hell? You're an RNA polymerase. Your job is to interact with specific DNA sequences and turn them into RNA, and you don't have DNA binding affinity? Huh? They don't. Instead, what these guys use is a whole range of proteins that are generically called transcription factors. And the transcription factors that we'll be looking at are given the name TF, and then whatever RNA polymerase they're working with. So you can have TF1, which nobody talks about. You can have TF3, again, nobody talks about. Or you can have a whole set of TF2s, which we're going to talk about. So we have TF2A, which is a real big one. TF2A is one of the critical interacting factors that is part of the transcription apparatus. And actually the big one we want to talk about is TF2D. You might ask, well, what happened to A and B and C? Yeah, they're there, they're just not as interesting. TF2D is the eukaryotic RNA polymerase II transcription factor, and it really is doing a lot of the heavy lifting. So a way to think about eukaryotic systems, we still have the same kind of vocabulary in terms of the promoter sequence, control region sequences. We still have +1, and we still have the structural gene. But now, instead of having sigma factor that allows the RNA polymerase to bind specifically, what we've got is the transcription factors, and they are doing the binding. So we've got TF2D, and within TF2D is something that is called the Tata binding protein. And TF2D has intrinsic DNA binding affinity. It is binding to promoters. And there are a range of different transcription factors. Let's see, I'm trying to think if there's anything that I need you to really worry about. We've got Tata binding protein, TF2D. TF2B is also floating around in here. So Tata binding protein is kind of a little ball within this. What we're doing, the way I think of it, is we're building a Pol2 nest. Pol2, and the RNA polymerases in general, they don't bind to DNA, they bind to transcription factors. The transcription factors are building this little bed, this nest. Pol2 then can bind here and carry out initiation. So, kind of like sigma factors aren't needed for elongation, the same as transcription factors. These aren't needed for initiation, for elongation. They're needed only for initiation all across the nation. No urination. So that's a similar kind of model there. But the really critical point is, without the transcription factors, Pol2 doesn't even hang out with DNA. And just from a sort of a philosophical perspective, I kind of think about this in that dramatic pause as I reveal a secret to the universe that everybody in the web should know. With eukaryotic gene regulation, particularly in complex eukaryotes, there are genes that if you turn them on, you will trigger cell division. And that could be a bad thing. You're not ready for it, you haven't replicated your DNA, you don't have enough energy to do it. You know, we only want to trigger cell division under very precise conditions. I don't want cells just dividing kind of like, woohoo! No. You divide when it's time, you divide this way, you divide that way. Ain't no monkey business with cell division. And there are a whole range of genes that we carry within us that if you turn them on inappropriately, you can really jack stuff up. So consequently, our gene regulation system is a lot more constrained. It is, we're talking loaded gun stuff here, and so you don't jack with like, oh yeah, we have binding affinity, the RNA polymerase can bind, because in bacteria, they are kind of drooling out all their genes all the time. 1 or 2 copies until something's really turned on, and it's like they're single cells. It's just not going to be that big a deal. It doesn't really get them into trouble. With you and I, a single RNA transcript from a specific gene could absolutely start a tumor. So it's like we got to be really careful about gene expression and have it under really tight control. Consequently, we don't even let RNA polymerase near our DNA unless this is the gene we want to transcribe right now, get to work. It's not like it gets to hang around and sort of play about. No, we do not take risks like that. So we produce an RNA. Now, when RNA polymerase comes in, RNA polymerase actually is a fairly big looking guy. The RNA polymerase actually carries with it a whole range of TFs on him as well. Not that we need to worry about that. What this also does is once transcription is initiated, there is a tail on one of the proteins of RNA polymerase, which is made up of a number of different proteins. So the tail on an RNA polymerase protein, and it's going to become phosphorylated, and that's kind of a signal to the system that, all right, everybody, I'm starting to transcribe a gene here. I need all these next stage processing proteins to gather around. And we'll see what these next stages are, because this is really and truly what separates out the eukaryotes from the prokaryotes. So we have on this carboxy terminal tail a range of different phosphorylation events that are saying transcription has begun, all aboard, and we start moving down the transcript. Now, why this is important is because the transcript, once it's generated, is not just left alone. And this is another really key difference between prokaryotes and eukaryotes. And so in terms of a list, this is like, yeah, you'll be asked this. Eukaryotes have RNA processing. If we were to do a comparison between prokaryotic RNA processing and eukaryotic RNA processing, that's a real easy one. In that prokaryotic RNA processing, the list is actually quite short in that there is none. Okay, I got that one. I got that question right. So, eukaryotic RNA processing, though, is a good bit more complicated. First, and these things happen kind of all at the same time, in that you have the addition of the 5' cap. This is a guanine that is actually laid on backwards. It's a phosphate-phosphate bond rather than a phosphodiester bond. It is referred to as the 7-methyl guanosine cap. So that's added on, and it's what we refer to in the biz as a template-independent nucleotide, i.e., there's not a G at the first nucleotide of the transcript. This is added on afterwards. So we have the 7-methyl guanosine cap. The second thing that is done once transcription has finished and the transcript has been terminated, we have the addition of the 3' poly A tail. Now, I'm having to think about this. This is pretty much all Paul 2. I have to look to see if 5' caps are happening on the other guys, but for our purposes, let's go ahead and say that no, this is strictly Paul 2. Guaranteed. Paul 2 is the only RNA polymerase that adds on the 3' poly A tail. So RNA polymerase itself isn't doing it, it's recruiting these different factors. And the poly A tail is like 300, 400 A's. A. So you have this really long poly A tail. That's a key difference right there between prokaryotes and eukaryotes. Prokaryotes, they terminate, that's it. Eukaryotes, the transcript terminates. We have the 5' cap added on the 5' end. We have this poly A tail that is zipped off on the 3' end. We now are beginning to process our RNA. Just as a little note, one of the ways that you can purify messenger RNA is to do something called oligo-D-deoxythymine chromatography. They make little chromatography columns that like that big. They have little particles of silica, little bits of sand, with a stretch of T's on them. You pass the RNA mix over this column. The poly A tail base pairs with the T stretch, and it'll hang there. And you can wash everything else off, and then you can elute or remove it from the column by putting a little bit of water in there, lowering the ionic strength, and the base pairing dissociates. And you can generate a reasonably pure messenger RNA population. So, all right. Now, these two additions are just slapping stuff on one end or the other. The third and final processing event is a bit more complicated, but nonetheless critical, and that is this idea of exon intron splicing. Just a little point. You guys remember this from genetics? Cool. Or not. Whatever. All right. So, and those of you in TV land, can you see that? It says exon intron splicing. Yeah, there you go. So back in the early days of gene expression technologies and things, when people were looking at eukaryotic RNAs, it was assumed that they were being processed in a similar way to prokaryotes, but then there starts to get this mounting evidence of, hey, the transcript that is generated from a given gene is often way shorter. What the hell is happening? What is happening is that parts of the RNA are being spliced out. And we refer to these as, well, the guys that first discovered these, and I really should know their name, I don't know who it is. Regardless, they refer to them as intervening sequences. And so, to kind of be hip, they refer to intervening sequences as introns. Now, what does that mean for the parts that are left over? The leftoverons or the remaindons? Well, they refer to the parts that are kept as the exons. So, we have introns, intervening sequences that are spliced out, and exons. And it works like this. We have a transcript. And now, because we're doing processing, this is referred to as the primary transcript, the unprocessed transcript. And this is a little point. This is all happening in the nucleus. This is before the RNA is being exported. So, the primary transcript is going to have this 5' cap, and I like to think of this, this inverted G is a really weird fist sitting out there. I think of it like a flashing strobe light that says, hey, this is the 5' end of a messenger RNA. It is a really weird structure sitting out there. And then we're going to have the addition of the poly A tail. And then we have splicing. So when we start thinking about splicing, what we can do is take a look at either the RNA primary transcript, or it's easier to look at the genomic DNA. That's primarily how we start doing this. You look at the genomic DNA, it might look something like this, where you know this is +1, and so this is the promoter sequence out here. And you look and compare what portions of the genomic DNA are found in the processed RNA. And very often it'll be diagrammed like this, where you've got these little boxes. And the boxes typically are indicating the pieces that are left behind after processing. These are the exons, and these parts are being spliced out. So we can have, and I have to do this awfully complicated, let's go ahead and put a couple of, here, I'll change colors. We'll make some spots here that we will think of as the exons. We'll keep the 5' cap. So these are going to get spliced out, these parts. They actually get looped out, and you have an actual reannealing or a covalent bond being produced to produce then an RNA transcript. 5' cap. There's a splice junction there. Splice junction there. And then the poly A tail. So you're not going to splice up the poly A tail regardless. And what we end up with is the processed messenger RNA is shorter, sometimes by thousands and thousands of nucleotides. There are some genes that have got more intron than there is exon. And so you produce this really long RNA, and then you splice out these parts, and you link it all together, and you have just the mature RNA. It's a little bit like, if you're playing with a bunch of trains, you can start pulling out individual cars, linking the other guys together. And this is the RNA processing event. What we're going to just throw in, but we'll pick up on Friday, one of the things that really makes this system sing is the notion, the idea of what's called alternative splicing, in that we can have the same gene produced in two different cell types. It can be alternatively spliced to generate, in rare cases, completely different amino acid sequences, completely different RNAs because of alternative splicing. Most of the time it's not that dramatic, but you have, this intron is present in this RNA, and this bit is spliced out. And so really, the term intron exon has to do with what the RNA is. One RNA's intron is another RNA's exon. Alternative splicing is a pretty cool trick. And we'll pick up on that on Friday. That's all I have to say about that. Rock on, everybody. Y'all be cool.